A'udzubillahi minasyaitonirrajim. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Let's continue with the second part of the cytoskeleton. It's me, Dr. Muhammad Hamzah Nawal Nasir, will be your instructor for the rest of the semester for SBT1322 Cell Biology. Alright, as you can see here, there is a QR code over here. You can actually use that QR code to record your attendance whenever you listen to the video of this slide. This week we are going to learn about microtubules. Compared to the microfilaments that you learned last week, microtubule is slightly bigger than the microfilament. Okay, between these three cytoskeleton, microtubule is the largest one. If you still remember, during the first session, okay, the part 1 for this lecture, I already mentioned about the size of microfilaments. Microfilament is about 7 nanometer, while the microtubule is about 25 nanometers. And microfilament, which you learned last week, is just a filament. Okay, there are no hollow structure of that filament. And this week we are going to look into microtubules. Okay, microtubules is differ compared to the microfilament by the structure itself. Okay, that's why we call it as microtubules, because you can see the tube structure, tube-like structure of this cytoskeleton. Here is the monomer, okay, the monomer of the microtubule. Okay, in microfilament, we only have actin. Okay, actin as the monomer. Okay, actin in the form of G form. Okay, the globular actin. While in this microtubule, we have tubulin and alpha tubulin and beta tubulin. Okay, over here is alpha tubulin and here is beta tubulin. Okay, these two monomer, okay, will form a dimer. Okay, we call it as tubulin dimers, and they arrange themselves so that they form a micro tube structure. Okay, it's just like a straw. If you can see here, they are arranged in this direction. Similar to the microfilament, microtubule also have the plus end and minus end. This will give a direction to the motor protein when they want to use the microtubule as a highway. And every single strand of the sequence of alpha and beta tubulin, we call it as protofilament. Okay, and the additions come from this plus end, while the dissociation or depolymerization of this microtubule occurs at the minus end. Here is the structure when we look at the complete structure of microtubule. And if you can see here, there is a hollow area. Okay, this can be seen even using this electron microscope. Okay, the transverse orientation of microtubule will show something like this, and you can see that there is a globular protein over here, and that is the alpha and beta tubulin. Okay, from the side longitudinal view, we can see that it has this lumen in the middle of the structure. Similar to the microfilament, the polymerizations of microtubules also involve the conversions of energy. Okay, in microfilaments, the conversions occurs between ATP to ADP. However, in this microtubule, the polymerization involves the hydrolysis of GTP, okay, to become a GDP. So that means the free form of dimer of alpha and beta tubulin, they have the GTP. Okay, that means it has a guanylyl triphosphate. Okay, the stabilization or polymerization of this dimer will form a strand of protofilament, and during this stage the hydrolysis of GTP occurs, and when they have the GDP, the protofilament will turn slightly curved. Okay, if you can see over here, it's slightly curved compared to this one. It's totally straight. Okay, curve because of uh, the twist of the protein structure in the beta tubulin. So it will cause the uh, alpha and beta tubulin, or this tubulin dimers, to turn, okay, and form a tube shaped structure. And once they have this protofilament, they also may undergo depolymerization by taking up the energy. So now, when they want to remove, that means they must have the GTP, okay, which later will come part of the pool of alpha and beta dimers, okay, alpha and beta tubulin dimers for the use of polymerization in other microtubule. This polymerization can be inhibited by using colchicine and colsamide. If you still remember that microtubule is important during mitosis and meiosis. So for us to capture the mitosis at the metaphase stage, we can actually use the colchicine and colsamide to inhibit the polymerizations and depolymerizations from occurs. With that, we can capture the sister chromatids, okay, the sister chromatid, and also the homolog chromosome aligned in the middle of the cell. Still remember about the microfilament? Okay, similar to that, the polymerizations of microtubules can also be controlled by the use of GTP cap. Okay, in microtubules, the growing and shrinking occurs by the polymerization and depolymerizations of the protofilaments, or it can also be the dimer itself, okay, the alpha and beta tubulin dimers, okay, and it shows that it has the less stable region of microtubule containing the GDP tubulin dimers, while over here they have the GTP, which is very stable. Remember that in the microfilament, they need the nucleation when they want to actually expand to polymerizing it. Okay, so in microtubule, they have the gamma tubulin. Okay, gamma tubulin for the nucleations of microtubules at the minus n. All right, so in the microtubule, the gamma tubulin will be the one at the negative end. Okay, because the additions of new uh, dimer, okay, the tubulin dimer, alpha and beta tubulin, uh, it happens at the positive end. Okay, so that's mean the length of this microtubule will comes from this side, so the length will go over there. Okay, and here is how we look at this microtubule during the nucleation, and you can see here it has a blunt end. Okay, it has a blunt end where the gamma tubulin, okay, the gamma tubulin over here to start or to become a nucleus for the polymerizations of alpha and beta tubulin dimer. Microtubule is not standing as a microtubule alone, not like microfilament. Okay, microtubule, it has several microtubule associated protein. We call it as MAPs. These MAPs are categorized into three main categories. The first is the structural MMPs, second, motor proteins, and the third one is enzyme and also signal molecules. Okay, in structural MAPs, okay, structural microtubule associated proteins, they are needed for the microtubule assembly, which link to the microfilament and also intermediate filament. The structural MMPs is important for the shapes and also the polarity of the cell. Okay, still remember about the polarity of a cell where it has a different uh, properties between the upper side and the other side of a cell. The example of the structural MAPs is the TAU protein, MAPS1 and MAPS2. The other microtubule-associated protein is the motor protein. Still remember about the transport of a vesicle during endocytosis and also exocytosis. We need the motor protein which slides on this microtubule for the vesicular transport. That means during exocytosis, it will bring out the vesicle away from the nucleus, while for the endocytosis, okay, exocytosis away from the nucleus, endocytosis, the motor protein will bring in the vesicle to the nucleus or to the uh, organelle needed for energy. Okay, now the example of motor protein is like kinesin and also dynein family motor protein. The third class for the microtubule associated protein is the enzymes and also the signal molecules. We need them such as the glycolytic enzymes and also kinases to be transported towards the other part of cell, and they are really important for the assembly of macromolecule. Here is the example of the structure of motor protein. We have over here is the kinesin, myosin, and also dynein. Remember about the exocytosis and endocytosis? The vesicle were actually carried by this vehicle and also this vehicle. This motor protein, they have the motor domain and they also have the stalk or the shaft for the connection, and also the associated polypeptides which actually link to the vesicle. So the area of motor domain is the one who actually attached to the microtubule. Over here is the diagram to show that the microtubule and also the associated protein. Okay, and the example here is the motor protein. As I did mention before, the microtubule, they have the minus N and also plus N. Minus N are located near to the centriole. Okay, while the positive N or the plus N located near to the membrane. which means the minus N is the one that located near to the nucleus compared to this membrane, which is away from the nucleus. If you look at this image, okay, the dynein and kinesin, these are the two main popular motor protein which uses the microtubule for the walk path. Okay, we can call it as a walk path or as the highway for this dynein and kinesin, because dynein and kinesin is a vehicle for the movement of vesicle, whether for the exocytosis, which means it moves from the minus N to the plus N, and also the endocytosis, which the motor protein must move from the motor, or sorry, from the membrane to the nucleus. Okay, from this function between the dynein and kinesin, we can see that dynein is the one who actually carrying the vesicle from the endocytosis, while kinesin is the one who actually bring out the vesicle from the cells towards the membrane for the exocytosis. Okay, and here is the example of electron microscope image for this motor protein. And if you can see here, they have the heavy chain and also light chain between the head domain and also tail. I would like to repeat, kinesin is the motor protein that move towards the positive end. Okay, which means they are moved towards the membrane. While dynein is the one who actually move towards the minus N, which is the nucleus. Okay, for you to memorize this easily, kinesin and dynein actually bringing you two different meaning. When you want the energy, okay, when you want the energy, that means you have a lot of energy. Okay, you have a lot of energy. In this case, you have a lot of CAMP. You will feel very active. With that, you will start to run toward the membrane. Okay, that's why we call it kinesin. It's a highly kinetic motor protein. When your body need energy, they will start to bring in the nutrient vesicle. Okay, the nutrient come from the membrane, okay, the outside in the serum, and then go to the membrane. Okay, from the membrane, it will bring to the factory. Okay, to the factory. So that means dynein is the one who actually bringing the nutrient towards the cell. Okay, the central core of cells. And for you to memorize this, this is when you have less energy, you need to eat, and we call it as dine in. Okay, you dine in your lunch, you dine in your dinner. All right, so here is the example of between the dispersions and also the aggregated of the pigment cells when the cell have the decrease CMP or increase in the CMP. All right, when the increase in CMP occurs, the pigment will disperse. Remember, when they have a lot of energy, the kinesin will bring the vesicle move towards the membrane area. While when the cells are at the lower energy, okay, lower CMP, so they will bring the vesicle towards the nucleus or towards the central core for the microtubule. We call it as mTOP. MTOC. Okay, microtubule organizing centriole. This is the cycle of kinesin, how it's actually gliding on the microtubule. Okay, here is the microtubule, and here is the kinesin shaft, and here is the motor head. Okay, here is the motor head, and if you can see here, the movement is controlled actually by the conversions between ATP and also ADP. Okay, this is ATP and over here is ADP. The arrow move like this. Okay, so that's mean the kinesins will actually turn its leg to the front by the conversions between ATP to ADP. Okay, and when they have the stage of ADP with ADP I, okay, then it will start to move the other side. Okay, the left leg and the right leg. Okay, and then the cycle continue. Okay, so that's mean the front leg always have the ATP. Okay, and for the right leg, it is the conversion between ATP to ADP. The microtubular system in the cells, the example is the centrosome. Okay, here is the centrosome or the centromere. The cilia and flagella, they have, here is the minus N and here is the plus N. The dividing cells, we do have the minus N at the core and the positive N at the outside. And for the vesicular transport, it is always happen in the neuron when they have to bring the vesicle with the neurotransmitter, okay, from the body of the neuron through the axon, okay, and then end at the dendrites of the cells, okay, of the neuron cell. And the minus N is at the centrosome and the plus N is at the near to the synapses of at the dendritic feet. Remember, I did mention about the mTOC, microtubule organizing center. Okay, microtubule organizing center, which is the negative end of the microtubule. All right. So the elongations, if you can see over here, the red arrow, the elongation is away the mTOC. Okay, away from the mTOC. And the mTOC have this kind of structure, 9 times 3 microtubule of A, B, and C. It has two centrioles at a right angle, and the organizer of it is the gamma tubulin in the pericentriolar matrix to form the minus N of forming by the microtubule. All right, here is the example of electron microscope images of this mTOP microtubule organizing center. All right, let's have a look at this cartoon. Okay, the diagram. Centrosome, they have the nucleating site for gamma tubulin ring complex. Okay, remember, gamma tubulin is very important for the nucleations of microtubule. That means that is the minus n of a microtubule. All right, and they have the centrosome matrix where they anchor the uh, the microtubules, okay, to that substrate. And over here we can see there are a pair of centrioles inside this centrosome. This is when the elongation or projections of microtubules away from the centrosome. Okay, here is the minus n, here is the plus n. This is the image from a confocal microscope to show the associations of this centrosome, okay, which contain pair of centrioles, and also the gamma tubulin over here. You can see the red color gamma tubulin. Actually, here is the centrosome matrix, and producing from it is the microtubule, and here is the positive end. That means this side is the positive end of a microtubule. Okay, let's have a look at the cilia. Okay, cilia in on a paramecium. How it moves? Okay, I just want to show you how it moves because motor protein and also microtubule is important component for the cytoskeleton, and it helps or assisting the cells to actually uh, helps the motility of a cell. Okay, and the structure of cilia is like this. Okay, it has the outer dining arm. Okay, so that means they use the dynein. Okay, but these dyneins are attached to this alpha and beta tubulin. And this alpha and beta tubulin actually form a dimers. Okay, form a dimer. And you can see here they have the alpha microtubule, sorry, not alpha, the A microtubule and B microtubule, which form a diprotofilament, and they have the dynein, okay, the dynein arm, and this dynein arm attached to this microtubule, which we know that when uh, there's a state of conversion between CAMP, okay, high and CAMP low, it will actually cause the rhythm movement of this structure. What are the molecules that compose the cilia? There are 25 types of molecules, but most of it, okay, about 70% are the alpha and tubulin. While they also have the dynein arm. Remember about the dynein arm, uh, which actually protruding from the A microtubule and form the uh, mechanism for peristaltic movement of a cilia. Not peristaltic movement, sorry. Let me correct it. It's a rhythmic movement of a cilia. Here is the role of the dynein arms in beating cilia. Okay, how this dynein arm actually causing the movement, okay, the beating cilia movement on the protozoa. Okay, they do have this positive positive and this is the negative end of that microtubule. All right, attached to it is the fixed attachment of diamine head, and the motor head is the one actually attached to the other strand of microtubule. Okay, A and B microtubule. So when they add the ATP, this microtubule will move to the bottom while this one move to the top. Okay, it's depending on the anchorage of this microtube. Okay, so that means one strand will move to the bottom, one will move to the other side, which will cause the stretch over here. All right, if they have the fixed anchorage at the positive end and fixed anchorage at negative end, for sure it will cause a bending of membrane at this side. All right, this is what we call it as telescopic effect because the additions in terms of uh, total length. Okay. All right. With this, when they have the linking proteins, remember about the attachment over here. Okay, and it will cause bending. This is how it looks under this cartoon diagram on the bend or the curve two microtubules between each other. Okay, which is the structure of a cilia. Microtubules is important for the mitotic spindle and also the attachment at the kinetochore. So that's mean at the kinetochore, the negative end, sorry, is a positive end, while this is the centriole is the positive is the negative end. Okay, so this is positive, and while over here is the negative end. All right, so that's mean during the metaphase, what will happen is the separation between the sister chromatids because of the shortening of this microtubule. All right, this is what happened. Still remember about they do have the spindle fiber. Okay, and they also have the interpolar microtubules. Okay, interpolar microtubules is the one actually attaching between each other so that they can hold and to maintain the distance between this and this. Okay, if they don't have this interpolar microtubule, what will happen is if the shortening of this and this microtubule, what will happen is the centriole will move towards each other. So they need another microtubule who actually maintain the distance between these two pairs of centrioles so that they can actually push between each other. Okay, so they are pushing between each other because here is the positive end, here is the positive end, and as you remember before, it will form like a arm that actually pushing each other. Okay, how motor proteins can organize the positions of cells organelle. Okay, this is how it looks. Okay, when the cells need energy, what will happen, or when they want to mobilize for the exocytosis, it will happen in this way. The vesicle will move away from this Golgi apparatus. Okay, because Golgi apparatus is the region where most of the macromolecules uh, being assembled. Okay, in the cell. All right, when the endocytosis, that means they will bring in the nutrient from outside, so it will bring in the vesicle to the area where the energy been produced. All right, so just repetition, dynein is the motor protein and it's carrying the vesicle, or you can call it as cargo, towards the towards the minus end. Okay, that is the dynein. All right, so let's continue to the intermediate filament. Okay, what are the intermediate filament? This is quite fast because it's very simple. Intermediate filament is a structure and specialized for bearing tension inside the cells. It's built from the keratin proteins, and that is a similar protein as our hair. So we call it as intermediate filament first because they are the one who actually cross-linking between the microfibers, sorry, microfilament to microtubules, uh, and also it has the intermediate in terms of size between this microfilament and also microtubules. Okay, microtubules is the biggest one. Microfilament is the thinnest one, while the intermediate filament is the intermediate, okay, the middle one. The function of this intermediate filament is to hold things in place inside the cells. So that's why they need to be not too big, not too small, but just enough for the cells to uh, hold the organelles in place. Okay, so more permanent fixtures of cytoskeleton, that means they are the one who actually cross-linking, reinforce the shape cell. Okay, the cell's shape and also fixed organelle location similar to this. Nucleus is held in place by a network of intermediate filament. This is what you need to know. That's why nucleus is always in the middle of a cell, because over here they have the the negative end, while the membrane they have the positive end. All right. Just want to show you the monomer of this intermediate filament is the alpha helican shapes, okay, in the form of alpha helica, and they will form the coil dimer, and this is when the parallel dimers occurs. Okay, anti-parallel tetramer, okay, will form more strength to the intermediate filaments, while the protofilament is the one who actually uh, the most strongest, okay, are the strongest microfilament, sorry, the strongest intermediate filaments in this cell, in a cell. All right, the mechanical characterization of cytoskeleton components, we can see that deformations and actin filaments. When the force come in, okay, when the force increase, okay, what actually resists the deformation, okay, the first part at the lower force is the microtubule, okay. Beyond than this, the microtubule will rupture. All right, and then it comes to over here the actin filament. Okay, actin filament, it can resist certain degree of force, but it cannot be as flexible as the microtubule, because uh, even at the higher uh, force, it's deformed very slight. All right, and it's easily being ruptured by the mechanical force. While the vimentin, okay, the intermediate filament, it has some sort of flexibility, and if you can see, there are no event of rupture. It shows that my intermediate filament is much more stronger compared to the microtubule and also the actin filament. All right, the intermediate filament important for the tissue specificity because it's buffer against the external mechanical stress. Okay, and it has, for example, over here in the epithelial, we do have the keratin, while in the connective tissue, muscles, and neuroglia, we do have the vimentin-like structure, just like vimentin, desmin, and also glia protein. The neurons or axon, we do have the neurofilaments because the structure of neuron is much more bigger compared to these cells. All right. Okay, the exceptions, nucleus, for example, the lamins A, B, and C, because they do have the lamina fibrosa. The domain structure of the intermediate filaments, okay, because you need to know this as well, because it has a head and also tail. Okay, just like in keratin. But vimentin, they do have the head, but for the tail, it is very short. In a neurofilament protein, it has a structure like this because they must be able to uh, stand with the changes in stress or mechanical stress of a neurofibers. All right, I will leave it for you to look at this because it's just showing how this cross bridge at the non-helical at the C terminus, and it actually giving an idea which cells need the intermediate filament the most by looking at the function of that cell itself. Uh, this uh, this slide showing you the muscle movement, okay, at the tissue level, skeletal muscle. For example, how this cytoskeleton works on the cell, okay, at the cellular level, or the collective migration, which means the cells move, okay, not the intracellular movement of the cell. Okay, I just repeat over here the movement of the cilia and flagellum, okay, in this motion. And how can they achieve this motion? Remember about the dining arm, okay, the dining arm helps the sliding, and they do have the uh, interconnecting plates, okay, the blue color over here, okay, over here the netzine, which actually helps the to uh, to give stress, okay, to give a stress and also the force to the cilia for them uh, to bend it a bit. Okay, in the amoeboid movement, we need the cells with this ability, and this can be only be achieved when they have this uh, cytoskeleton. Okay, and over here I just want to show you about the composition of the thick filament in the sarcomere of the motor protein, and they are involved in the contractions of a muscle. Okay, the contraction and relaxation of muscle. So okay, here's the sarcoma when we can see that on the skeletal muscle. Okay, because they have uh, the filaments, okay, at thin filaments, and also they have the myosin at the thick filament. Okay, at the myosins it has the motor protein, okay, that we call it as the myosin head. Okay, myosin head. Okay, this area. So these things will actually attach or gliding on the thin filament, which will causing the sliding. Okay, not okay, sorry, not this way. It's sliding of this area and this area towards the M line. Okay, so when this myosin head pulling this actin or thin filament, it will actually pulling the Z disc move closer to the M line. Okay, so the total length of this skeletal muscle will be shortened. It will give the impact of muscle contraction. Alright, so here is the example of it for the motor protein of myosin. And okay, just look at this. Here is the cartoon where I did mention about when the myosin bring it or pulling the actin filaments, okay, the thin filaments, and it will actually decrease the M line, okay, the dark band, to become a contact muscle. All right, so just repetition over here, showing that this the disabilities or the abnormalities of those protein, okay, in the DMD, Duchenne muscular dystrophy, or DMD, uh, which shows that the muscle have a difficulties in terms of movement. Okay, that is just because of the myosin head. I think uh, just to recap, we have learned about the cytoskeleton. Okay, I want you to focus more on the cytoskeleton and also the associated protein, but don't too details in terms of the motor proteins. What you need to understand is this microtubule, some of them works as the highway. Okay, some of them work as the structure for the cilia and flagella, while the microfilaments, it works for the cell divisions, and these intermediate filaments for them to actually keep the tensile strength of a cell, as well as the positioning of an organelle. I think that's all from me for this week. If you have any question on this, you can actually ask me in the Telegram group. Inshallah. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.